we're going to start looking at the neuromuscular junction. It's kind of jumping ahead a bit to the nervous system, but we need to, it bridges the muscular system and the nervous system, but we have to kind of dive into it to understand what that connection is and how it brings about the contraction that we just talked about, because we keep saying that this impulse, this nerve impulse, is what's causing the contraction to happen. So we need to look at our neuromuscular junction. By definition, a neuromuscular junction is going to be that spot where the neuron and the muscle come into contact with each other. If you think about you're driving down a road and you're looking for a new road, there's a junction. That means it's going to meet up with another new road. The neuromuscular junction specifically is a synapse between a motor neuron and a muscle. A synapse is a connection. Okay. Now keep in mind, there is, and we'll talk about this several times, there's a physical space between the neuron and the muscle, but they are in close contact with one another. They're in close proximity with one another. Specifically, we're talking about, there are different kinds of neurons. We're talking about a motor neuron. And if you think about what a motor does, it causes movement. So a motor neuron is going to be a neuron that's going to be associated with the muscular system, and that's the case. So when we talk about our synapse, we've got our neuron, this motor neuron that we're talking about. We are most interested in the part of the neuron called the presynaptic terminal. Pre means before, so we're interested in the part of the neuron that happens before the synapse. Okay, so that end. I've referred to it in previous lectures as a gecko toe. Contained within the presynaptic terminal will be synaptic vesicles. We know that a vesicle is one of those membrane-bound little bubbles that we're going to store things in. In this instance, it's going to contain a neurotransmitter. Specifically, the neurotransmitter is acetylcholine. From this point, we'll refer to acetylcholine as ACH. Acetylcholine is going to be the neurotransmitter, the only neurotransmitter that we're going to talk about with relation to the muscular system. When we get into the nervous system, there's going to be lots and lots of different neurotransmitters. But for right now, we're only going to concern ourselves with acetylcholine because that's the only one that has anything to do with the muscular system that we're interested in at this point. After we finish with the neuron, we have our presynaptic cleft, our synaptic cleft. Synaptic cleft is that physical space between the neuron and the muscle. In this instance, we're talking about a skeletal muscle. We refer to the piece that we're interested in as the motor end plate. It's going to be part of the sarcolemma, part of the plasma membrane of the skeletal muscle cell, and it contains receptors for neurotransmitters. A receptor is going to be a protein molecule that is built, its shape, to accept a specific molecule. Allow that specific molecule to plug in. In this case, that specific molecule is acetylcholine. This is kind of a close-up of our neuromuscular junction. The yellow broadened piece is the gecko toe, or the presynaptic terminal. The red line, as it has been in other diagrams, is the sarcolemma. This specific piece we would refer to as the motor end plate because that's going to have molecules, receptor molecules, that will allow the acetylcholine to fit in. You'll see that we have in our gecko toe, in our presynaptic terminal, we have synaptic vesicles that are filled with teal color balls. Those teal color balls are representing the acetylcholine molecule. And as the synaptic vesicle dumps the acetylcholine into the synaptic cleft, it's going to move across through diffusion to the motor end plate. As the nerve impulse reaches the axon terminal, the gecko toe, the synaptic vesicles move to the presynaptic terminal. They move to the end of the gecko toe. The synaptic vesicles merge with the neuron cell membrane in the process that we've called in the past exocytosis, right? So our vesicles, our synaptic vesicles, are going to merge with the membrane of the neuron, thus releasing the acetylcholine into the synaptic cleft. The acetylcholine will diffuse across the cleft to the receptors on the sarcolemma, to receptors on the plasma membrane of the skeletal muscle cell. This causes the impulse to travel to the muscle, causing the muscle to contract. Okay, it's going to cause that impulse to, it's going to cause the sarcolemma to be kind of quote unquote turned on and have that impulse travel along the sarcolemma and then down through the T tubules, causing our muscle to contract. As long as acetylcholine remains in the cleft, the muscle is going to be stimulated because it's hooked into the receptor molecule. And the whole point of this, how we got this whole thing started, was the acetylcholine diffused across and hooked into our receptor molecule. So as long as that ACH is plugged into our receptor molecule, the muscle is going to be continued to be stimulated, causing it to stay contracted. In order for this to stop, right, in order for this to cause our muscular contraction to stop happening, we have to break down the acetylcholine. In comes acetylcholine esterase. If we look at our last three letters, they are ASE. When we have a chemical name that ends in ASE, what is that telling us? That it is, in fact, an enzyme. What would the substrate be that acetylcholine esterase is going to work on? That's going to be our ACH. Acetylcholine esterase is found in the synaptic cleft. It's just kind of hanging out there. That's where we need it. That's where it's going to live. It inactivates acetylcholine by breaking it down. In actuality, it kind of chops the molecule in half into an acetate and a choline molecule. Nonetheless, once I break it apart, it's no longer acetylcholine, so it's no longer going to be able to fit into that receptor molecule and cause a contraction to occur. So in order for muscle relaxation to happen, in order to have the reverse of a muscle contraction, we need two things to happen. And they're going to involve two of the players that we've been talking about right along. First is acetylcholine. We've got to break it down. That's why we just talked about acetylcholine esterase, because it's in the synaptic cleft, and eventually it will start breaking down the acetylcholine, breaking it in half. This is going to cause the calcium ion release channels to close. It causes the calcium ion release channels to close because we've broken that connection between the receptor molecule and the acetylcholine, so the impulse is no longer traveling along the sarcolemma and down through the T tubules. When that signal is no longer being moved along that system, the doors are going to close. The calcium ion release channels are going to close. So that's the first thing that has to happen. We've got to get rid of the acetylcholine so that the doors can close. Once our calcium ion release channels close, we then have to start moving the calcium ions back where we found them. We've got to rip it away from troponin, we've got to rip it away from those myofibrils and actively transport, actively move it back into the sarcoplasmic reticulum. We have to do that because that sets up our stage for our next muscular contraction. So we've got to remove those calcium ions and put them back where they belong. Kind of like if you're having a party. Right? Mom's out of town, dad's out of town, you're having a party, you move the furniture around. Well, before mom comes back, you've got to put that furniture back where it belongs. So we've got to get those calcium ions back where they belong. We've got to move them back into the sarcoplasmic reticulum where they belong. As the calcium ions are removed, it's going to separate from the troponin. The troponin is going to go back to its normal shape, which causes the tropomyosin to go back to its normal shape, which causes the active sites to be recovered. The bottom line, folks, is just like if you had a party and mom and dad are out of town and you're not supposed to have a party, anything you move out of the way, you've got to put back exactly where you found it. So in order for the muscle to relax, we've got to take away anything that we brought in. So the calcium ions are going to get taken away. Well, when the calcium ions get taken away, the troponin changes its shape. When the troponin changes its shape, the tropomyosin goes back to its original position, which covers the active sites on the actin. Once those active sites are covered, no further cross bridges can be formed until the next impulse comes through. right? And again, these are the same diagrams. We're just going to kind of switch them. This is a stimulated muscle fiber. I know it's stimulated because the tropomyosin, has moved. My active sites are exposed. The myosin has bound to those active sites. Now, what's going to happen once we take the calcium out of there? Troponin is going to go back to its original position, which causes tropomyosin to go back to its original position, which covers those active sites. So the myosin head can no longer connect to our active sites. This brings us to a conversation of rigor mortis. I find this to be one of the most fascinating things we talk about easily in 131, perhaps in both 131 and 132. Rigor mortis is something that happens after death. It's that rigidity. The muscles become rigid. Well, how does that happen? After death, as the muscle fibers begin to break down, as the body begins to decompose, calcium ions leak out of the sarcoplasmic reticulum. Well, here's the problem. There's no energy available to pull those calcium ions back into. We can't actively transport them because we're dead. We're going to run out of oxygen eventually. So as the calcium ions leak out of the sarcoplasmic reticulum, they're going to bind to the troponin, causing the tropomyosin to move, allowing cross bridges to form. Once our cross bridges form, our myofilaments slide. We cause a muscular contraction. No further ATP is going to be produced because, again, the person's dead. We're not bringing oxygen in. So what's happening? Because you might be thinking, well, wait, why is any of this happening if we're not producing any more ATP? Well, remember, we've got storehouse of ATP, right? We build it up and we store it, we hold on to it. We also have some oxygen that's going to be stored in the myoglobin, which will allow ATP to be formed for a while. Eventually, that oxygen is going to run out. At that point, no further ATP is produced. Those cross bridges cannot be broken. You'll remember the cross bridge, the myosin bound to the actin active site. The only way that myosin let go was when we gave it something else to hold on to, right? Remember the toddler in the grocery cart, right? The only way that toddler is going to give mom the keys is if mom gives that child something else. The only way myosin was going to let go of the active site was if we gave it an ATP molecule. Well, we don't have any more ATP to give. So those cross bridges cannot be broken. The muscles are going to remain rigid or contracted, in other words. Now, rigor mortis follows a very predictable pathway. It begins about 3 to 4 hours after death. And again, we said it wasn't going to happen immediately because we have ATP stored, we have oxygen stored in the myoglobin, so we have a little bit of a savings account, if you will, of ATP. So it's going to take a while for us to run through that, and that's why it only starts about 3 to 4 hours after death. We reach maximum stiffness about 12 hours after death. Right? We've run through all of our ATP, all of our oxygen. We're maximally contracted, and it's going to start to dissipate. It's going to start to lessen about 48 to 60 hours after death. Please don't think that a dead body starts to manufacture ATP again. It does not. It does not start bringing oxygen in. The reason it starts to dissipate or lessen is because the normal decomposition process of a dead body, all right, that the cells just start to break down and everything starts to let go. All right, so that's rigor mortis, and this may answer the question if you watch, Law and Order or any of those kind of crime shows. They can tell, they look at the body, it's been dead for X number of hours. This is how they know, by how far rigor mortis has proceeded in this